Hello everyone, welcome. Hopefully everyone managed to get some lunch. I'm Dave Ritchie, I'm a haematologist and head of the bone marrow transplant service at the Integrated Haematology Service, which incorporates haematology here at Peter Mac and at the Royal Melbourne Hospital, both major clinical partners of the VCCC. We're very grateful for the VCCC and the staff for organising the lunch and making an opportunity for us to hear some excellent research from David Curtis, who I'll introduce in a second. For those of you who are regular attenders or who may even work in this building, you'll understand that the VCCC is more than just this building. In fact, at the very least it's two buildings, because there's a fair chunk of the building across the road as well. But organisationally it incorporates major clinical care providers in cancer therapies, incorporates world-class research, both here at Peter Mac, at WEHI, University of Melbourne, and at a number of other non-campus partners who are a little too many to mention individually. But it's a major collaborative exercise, it's a major organisational exercise, and the research that is so important for the future of cancer outcomes is taking place here and now, and part of that research, of course, is that which is being done in other major academic institutions across Victoria, including at the Monash University, where our guest speaker is from today. It's been my pleasure in the past and currently to work and collaborate with Associate Professor David Curtis, where we have a number of projects underway together and hoping to continue that collaboration with some novel clinical trial initiatives, NHMRC grants hopefully going our way. David has a long association with the Parkville area. He was a haematologist and a bone marrow transplant physician at the Royal Melbourne Hospital, and that's the opportunity that I had to work with him in the past. He did his PhD at the WEHI, and he has done substantial work through the Bone Marrow Donor Institute research laboratories, which were based at the Royal Melbourne Hospital. And he left for those labs and took up his current position as the Director of Malignant Haematology Research at the Australian Centre for Blood Diseases. And you can see his affiliations on this introductory slide. So of course he has academic positions through Monash University and clinical positions as a haematologist and bone marrow transplant physician at the Alfred Hospital. His research program is primarily focused on blood diseases, using mouse models to understand those better, both genetically and epigenetically. And he's going to talk to us today on killing leukemic stem cells, which as a haematologist is a major challenge for us, and really what stands between the progression of leukemia and cure. So join me in thanking both the VCCC and David for coming and speaking with us today.
Thanks very much, David, and thanks to the VCCC for inviting me to speak today. So the title, I was asked to give a sexy title. So I gave a sexy title, but I'm not really going to talk about actually killing leukemic stem cells. What I'll predominantly talk about is work that I've done in collaboration with a number of the people in this audience related to the development of a new small molecule inhibitor of an enzyme called PRMT5. But hopefully it will kill leukemic stem cells, and so I'll show you a little bit of data which suggests that that may be the case. So just briefly, I just wanted to actually tell people on the north side of the Yarra, if they're not familiar with the ACBD, it's a tough an acronym. It should be ABCD, shouldn't it? The founder actually thought he wanted to make it really, sound really important, so he called it the Australian Centre for Blood Diseases. But really, we are a centre of Monash University, and it's affiliated with the Clinical Haematology Department at the Alfred Hospital. And really, over the last 5 years, we have expanded a lot. We've been supported fantastically by the Dean of Medicine at Monash, who happens to be a haematologist, which always makes it a little bit easier. And now there's over 100 researchers and 15 lab heads with 8 clinician scientists, and I think that's one of the great strengths of our department, is that we've got clinicians who are doing a lot of basic research as well, and really I think that we've developed a great team. Because we're a clinical department, we're disease focused, and as you can see, we have a very large number of phase 1-2 clinical trials, probably the largest number of trials in Australia in haematology, and we run the national registries for AML and myeloma, and have a very large cohort of tissue samples. And very recently we developed, we got a successful grant from the ACRF to develop a blood cancer therapeutic centre. These are some of the PIs that work within the blood cancer division. Some of them I'm sure that you would recognise, and they're the diseases that sort of they're focusing on. And this was the ACRF grant, which essentially is a facility which is primarily focused on screening of fresh primary samples. Over the years, we've really done a lot of work to compare frozen versus fresh samples, and really the answers you get out of fresh samples are very, very different to what you get out of frozen samples, and certainly very different to cell lines, as many of you are aware. And so we're very focused on developing the infrastructure to have a seamless sort of transition from the clinic, getting the samples, processing and analysing them. And we're predominantly looking at high throughput drug screens. So we've recently got an IQ screener and have started doing a lot of high throughput drug screening of primary AML and myeloma samples. So for people who are interested in that, we're certainly open to collaborations to develop that further. So the leukemic stem cell is something that I've really been studying for the last 15 to 20 years, and it really stems from my interest in stem cell biology and hematopoietic stem cells that are used for bone marrow transplantation. And really it has become very clear over the last 20 years, particularly with genomic studies, that certainly for AML, likely for ALL, there are these rare cells which are the cells that are responsible for disease relapse. And so really I think, as David mentioned, one of the fundamental things that we need to try and sort out and overcome is the ability of these leukemic stem cells to avoid our current therapies, and really that's been the focus of my laboratory's work, and I'll tell you a little bit of a story about that. So what are the mechanisms of stem cell resistance? These are mechanisms which probably are retained by normal stem cells as well as leukemic stem cells. The top one is quiescence. We've got a large program which suggests that quiescence probably is a fundamental property of leukemic stem cells, and we're trying to sort out ways of overcoming that to eradicate them. There are a list of other ones which again are probably retained by normal stem cells as well as leukemic stem cells. So really that does raise a major question. Is there going to be a therapeutic window? Can we find therapies which are able to eradicate leukemic stem cells which have similar properties to normal stem cells? And that really is probably at the crux of many of our current therapies, and the therapies that we need to develop is we need to develop systems to be able to test that question so that we can then test them in the clinic. Now, not all is lost, is that I think many of us actually forget that chemotherapy does cure a significant proportion of patients. And so obviously if leukemic stem cells are present in all leukemias, but some people are cured, there clearly is a therapeutic window where the normal stem cells do work. So I think that does suggest that we will be able to achieve therapies which can eradicate these leukemic stem cells. And probably the simplest way to think of it is this concept of synthetic lethality. And that is where mutations that are causing the leukemia make them also more dependent on specific pathways. And so those mutations might cause a mutation-specific lethality, so that you need to direct the therapy to a specific for a specific mutation, or alternatively, they might be generic. These might be just generic things which happen to cancer cells, which make them more susceptible to specific types of therapies. And that's really what we've been working on, is to try and identify these generic sorts of dependencies. Very briefly, this is the sort of model that my lab uses. I personally think it is a very powerful model. We have very few models to study leukemic stem cells, and so we've really been focusing on transgenic mouse models which recapitulate human disease and are able to you can isolate the leukemic stem cells at various time points in the development of leukemia, as well as after treating these leukemias with our current therapies, to look at the types of leukemic stem cells that avoid the therapy. So I won't talk anything more about that model, but that's a really neat model, and again, happy to speak to people about collaborations to test, if they've got a specific pathway or a specific drug that they'd like to know, does it target these leukemic stem cells? Would love to know. And I should say that I don't think that it's the oncogene that really matters. People say, oh no, it'll only be specific for that oncogene, but I think a lot of these processes that make leukemic stem cells susceptible will be true of irrespective of what the oncogene is that's initiating the disease. So the story I wanted to talk about was this one about this generic susceptibility of cancer stem cells, and that's to do with ribosomal biogenesis. This was recognized about 120 years ago, that cancer cells have these prominent nucleoli, which are the factories that make the proteins for the cell, and they really distinguish the cancer cell from a normal cell. And really, over the last 120 years, I don't think actually much has really been understood or about how that is different. There is starting to be some understanding of how oncogenes like MYC can drive ribosomal biogenesis, but I think really we are at just the beginning of understanding this process of how cancer cells with these abnormal nucleoli that have a lot of ribosomal biogenesis are more dependent on that process. And I think actually one of the really nicest examples is this last line here is about tumours and MYC. So many of you know that MYC is a prominent oncogene and it can drive tumours, but if they are deficient in some of the ribosomal proteins, so if they have ribosomal deficiency, in animal models, MYC doesn't drive tumours. So you really do need an appropriate ribosomal apparatus to be able to develop tumours. So my disclosure is, for the data that I'll present, is that I'm a part of the Monash is a partner of the CTX CRC, and so we do get licensing fees and milestone payments from Merck from that, and I'm also on the Joint Scientific Committee for the CTX Merck collaboration, and do receive research funds. And so there are really three parts to the talk that I wanted to briefly discuss. One was the actual development of these inhibitors, and I think the story that sometimes you don't have to be first to actually have a successful story. So for all you researchers who you see it, you see some work that is similar to what you've been working on published, that doesn't mean that that's the end of your story, and that often you can actually use that as leverage. So the story actually starts with a non-hematologic malignancy with a blood disease called sickle cell disease, and it's a long-standing collaboration that I've had with Steve and Jane, and essentially it's working on an idea called fetal globin switching. So when babies are born, and develop at about 3 to 6 months of age. They switch from a fetal type globin to a adult type globin, and patients who have sickle cell disease, they develop the disease at 3 to 6 months of age because they've switched from that fetal to the adult globin. And so the idea would be if you can maintain the fetal globin, you'll be able to cure the disease. And so Steve had been working on this for about 20 years to try and work out what the regulators are of this fetal globin switch. And he identified an enzyme called PRMT5, which is in the complex that sits on the fetal globin promoter and basically silences it at a 3 to 6 months of age in humans. So the idea was to develop an inhibitor of this enzyme to prevent fetal globin switching. And so that was the idea. The problem was there was no really body to do that. We didn't have a commercial partner that was interested in developing this for a relatively rare disease in humans, or not in humans, but rare disease in Australia, very common elsewhere. And so we really struggled for a long time to work out how we could actually develop a small molecule inhibitor. Briefly, PRMT5 is of relevance, is an enzyme which was first identified as a binding protein for JAK, and it's ubiquitously expressed in stem cells, and it's the only enzyme that catalyzes symmetric dimethylation of proteins. It's not that simple because the modifications that it does induce in the histones can cause activation and repression marks. So you know it could do one of both things, activate and repress gene expression. But more importantly, it's not just an epigenetic regulator. So I'm sure people who work in the epigenetic field probably recognize that a lot of the epigenetic enzymes that they work on actually do modify other proteins. And so for PRMT5, it's well known that these proteins that are modified include the SM proteins, which are important for splicing, and ribosomal proteins, as well as some other important cancer-related genes. So we didn't have a commercial partner at that stage, and so we gave this project to a PhD student, and I think it was probably blind ignorance rather than being bold, but to his credit, he did develop a relatively facile screen, and it was that screen which identified a couple of small molecules which were able to inhibit PRMT5 to some extent without inhibiting other PRMTs. So they obviously weren't great small molecules. They weren't stuff that we could actually test. Even in vitro it was going to be difficult because they weren't potent enough. But around about that time, around in the late 2008, 2009, there started to be quite a little bit of literature evolving which suggested that PRMT5 was overexpressed in a lot of cancers. And so why they're overexpressed is still not understood, but simplistically you could think that if PRMT5 was important for this ribosomal biogenesis process, that maybe increased expression might be an advantage for cancer cells. But I guess the most important thing was that this link to cancer then actually got interest from the Cancer Therapeutics CRC. So I think all three people in this audience? Yes. In fact, they're all sitting together. There you go. After a lot of persistence from Steve Jane, I think Ian Street would agree that he was very persistent, and eventually convinced Ian to take on this project as for one of the projects of the CRC. And really, they did an enormous amount of work. Many chemists screening a large number of small molecules did actually identify a couple of very potent and specific small molecules, which was incredibly exciting. And here's some of the data for the 2 series that they developed, just showing that it is very specific for PRMT5 and doesn't inhibit any of the other enzymes. And pleasingly, the biochemical potency, even in cell potency, was very potent. So you can see for CTX034, 5 nanomolar potency, which really is sort of at the range that you would need for a clinical compound. Importantly, one of the critical developments with this drug program was being able to do crystal structures. So we were able to identify how this small molecule was working, and essentially PRMT5 has 2 pockets, one which has the methyl donor and the other has the arginine that is the substrate for the enzyme, which is the arginine within that protein. And the small molecules that we developed were ones which sat in that arginine's pocket rather than the methyl donor, and that gave it the specificity to work, and really was very critical in the eventual licensing of these small molecules. So this is the story about this is the rabbit. Is this the hare? The hare. Is it the rabbit or the hare? The hare. This is the hare. This is the epizyme. So unfortunately, right about that time that we were ready to publish the patents, another company had identified almost identical small molecule, which apparently is almost unheard of for two groups to come up with exactly the same thing in a screen, but unfortunately they did, and that really caused a lot of tears. However, if you think about it, they published a paper showing that these were really of some value, and so that's just some in vitro data, or no, some in vivo data showing that it is able to target mantle cell lymphoma, showing that it does work. And probably more importance was that Epizyme, who'd licensed this to GSK, meant that other pharma wanted it. If GSK had one, we need one. And so really that was, I think, one of the critical sort of turning points that developed a lot of interest. However, as I've said, we didn't actually have one that we could license. However, I didn't realise this, but the chemists have been working behind the scenes to develop some other compounds which were of a similar mechanism but were patentable. And they developed at least 3 series, maybe I think there's 4 now, which have been patented. And I can only show you, just for commercial reasons, some data related to CTX293. And as you can see, it's even more potent than the CTX034, which GSK had developed or patented or licensed. And so that gave us some hope, and also obviously gave interest to other commercial entities that we had something that which was going to be valuable. And this led to the licensing agreement, which really was obviously, I think, one of the highlights for many of the people working on this project, and probably something that, you know, very few people actually get the joy of being a part of, and that has been a very exciting thing, which was licensed last year for an upfront fee of $15 million US, and then some significant milestone payments if we can progress these small molecules. And that's really an ongoing project. This is just some data for people who don't work in blood cancers, just to show the sort of spectrum of activity of these inhibitors. So the higher up it is, the more sensitive these cancers are. So you can see that blood cancers are actually the most sensitive tumours to these inhibitors, but there are a broad range of cancers which are going to be likely sensitive to these small molecule inhibitors. So So that's the drug development. And so I just wanted to talk briefly about some biology. I'm a biologist, I'm not a chemist, so this is really the part that I've really enjoyed and my lab's really worked on very hard for the last few years. And it really revolves around an unexpected result. So for the junior people, I think one of the things that I have learnt over many years is that some say that I'm too much of a cynic, but I really don't believe anything I read. Because most of it is not true. And this is a perfect example of that. So don't be despondent if somebody publishes something and you go, oh, that doesn't fit with what I'm doing, because that might be because what they've published isn't actually correct. So that's the unexpected result. Oh, there's another unexpected result. And so this is related to myeloproliferative neoplasms. So these are blood cancers which are driven by a mutation or an activating mutation of JAK2. And if any of you are still awake, you might remember that I mentioned at the start that PRMT5 was first identified as a binding protein for JAK2. So that really, we thought, oh, okay, so there may be something to do with the PRMT5 and these myeloproliferative neoplasms. And I think from a clinician point of view, I think this is a very important disease that we really still don't have any great treatments. JAK inhibitors have made some difference, but it's predominantly just controlling the symptoms of the disease, and they don't really cure the disease, and that's because they don't eradicate the cancer stem cell that drives myeloproliferative neoplasms. And so the unexpected result came from this work, which was published in a reputable journal, Cancer Cell. So lots of people try and get their cancer research published in these journals. I hope there are no authors in this for this one. So essentially they published work which suggested that JAK2 inhibited PRMT5, and that inhibition of PRMT5 was important in the development of the disease. So an editorial that was linked to this paper suggested that we needed to find ways to activate PRMT5 as a therapy for myeloproliferative neoplasms. Now, I mean, the paper looked okay. However, it didn't actually make any sense. And the reason it didn't make any sense was that all the work that we'd been doing when we'd been testing these inhibitors was that it would suppress erythropoiesis, so red cell production. These guys are saying that loss of activity was increasing red cell production. And really, I think the most obvious example was we've got conditional knockouts of PRMT5. So if you knock it out in development, the embryos don't survive past probably E6. So we've got conditional knockouts, and when we knock that out, particularly even in red cell development, you don't get any red cell development. So our data would suggest that PRMT5 is critical for the formation of erythropoiesis. So furthermore, if you look at the stem cells, the sort of stem cells of patients with myeloproliferative neoplasms, either in JAK2 mutant ones or even in ones that don't have the JAK2 mutation, if you look at the mRNA expression of PRMT5, it's actually increased about a log, so tenfold higher in the stem cells of these cancers. Again, supporting this idea that the cancers need to upregulate PRMT5 in a response to being able to grow a lot. In contrast, another one of the PRMTs, and if you look at all of the other PRMTs, they're not increased at the message level, suggesting that there is some specificity. And so certainly we've developed some data which does suggest that PRMT5 inhibits JAK2 mutant red cell production, and it's a lot more potent than on normal red cell production, suggesting that there may be this therapeutic window. probably the strongest data that we've developed that PRMT5 inhibitors might be a valuable therapy for myeloproliferative neoplasms is been resorted back to the mouse models. Mouse models are particularly valuable at trying to work out the mechanisms of action. And if you look in the myeloproliferative or JAK2 mutant mouse model, again, if you look at the stem cells, they have a much higher level of PRMT5 in the stem cells compared to normal ones. And if you do a Western blot on the sort of immature cells within these mice and look at a typical mark that is induced by PRMT5, you can see that it is increased compared to the mark within normal cells. This is the experiment which essentially compared, we did a comparison with the ruxolitinib, which is the JAK inhibitor which has been licensed and for treatment of JAK2 mutant disease. And essentially the bottom line is that it works as well, if not better than ruxolitinib as an inhibitor for myeloproliferative neoplasms. The question really is whether, unlike ruxolitinib, does it suppress the malignant or the cancer stem cell that's driving disease, because that's really the fundamental thing that we need to overcome. And I don't have any strong data yet. We're still working on that, but I think one of the really nice things, if I could work out how to use this without doing that, it's all right. Oh God, help me. If you look at the plot here on the right lower side, this is the effect of the inhibitor or ruxolitinib on P21, which is a marker of activation of P53. So activation of P53 is how a lot of chemotherapy works in killing cells. And you can see that unlike ruxolitinib, you get a very high level of P21, suggesting that we're activating P53. And really that's one of the, I think, key ways of trying to eliminate these leukemic stem cells is to be able to, activate P53 for killing them. Importantly, it doesn't suppress normal erythropoiesis, so if you give this to a normal mouse, it doesn't suppress red cell production. Again, an important thing to show that there is a therapeutic window. And so the final bit that I wanted to talk about was how this is working, and this will bring us back to the idea about targeting the ribosomal biogenesis, which I think is a powerful mechanism to try and eliminate leukemic stem cells or cancer stem cells. And so this data is really generated from human cells treated with the inhibitor and taken 2 to 3 days after treatment, and then we do gene expression profiling and protein studies to try and identify what is the mechanism of action. So the first thing we could show was that it does activate P53, which is good. That's just some data demonstrating that it activates P53. However, it's still able to kill cells even in the absence of P53. So on the left-hand side is a plot just showing some Isagenix cell lines which are lacking the key downstream killers of P53. And you can see that in the absence of P21, even in the absence of P53 or purim and noxa, these cells are killed. They're not killed quite as quickly. So you can see at day 5 there is a difference between the, say, the purim and noxa knockouts and the wild type. But by day 7 of treatment, it didn't matter whether they had P53 apparatus intact or not. So I think that is important, particularly for solid cancers. I think these inhibitors will be able to work in the absence of P53. So I don't think that, even though it's a mechanism of action, I don't think it's critical. We were wondering how these inhibit JAK2 mutant red cell production, and if you look at the gene expression profiling, they certainly don't inhibit cytokine signaling. In fact, they look like they activate cytokine signaling, which we don't really understand why, but they do seem to at the gene expression level. But what they do is essentially suppress metabolic pathways. So these inhibitors appear to basically starve the cells to death. And you do see some evidence of autophagy in cells that are treated with these inhibitors. But if you look at the gene expression profiling, it really has a very strong profile for starvation. And this is despite these inhibitors actually activating the MTOR pathway. So the MTOR pathway is really a fundamental growth signal that many cancers have. And there's a lot of work testing inhibitors of the MTOR pathway, but unfortunately they don't work that well because there are other compensatory mechanisms to get around that, that signal. These inhibitors actually do, in some cells, activate the MTOR pathway. However, if you look at the plots down below, it actually shows that they induce a cell signal or a signal that looks like rapamycin, so it looks like an mTOR inhibitor. So the pathway is being activated, but the actual response to that pathway is actually being blocked completely. Again, suggesting that maybe they are working at that ribosomal part of the mTOR pathway. And I think this is probably most beautifully demonstrated by the ability of these inhibitors to inhibit a MYC signal. So MYC is the classic sort of mTOR oncogene which activates cell growth, and these inhibitors basically suppress many of the MYC targets, these growth signals. So you would imagine, based on this data, that certainly tumours that are driven by MYC should at least have some sensitivity to these inhibitors. How's it doing it specifically? Well, we've generated a lot of data to show that it inhibits protein synthesis. And some really nice work done by the Hannon lab, who have now moved to Canberra, essentially show that it disrupts the ribosomal assembly. So it sort of prevents the 40S and the 60S from coming together, and so you get a lot of, ADS, which then can't actually get onto the polysome. There are three ways that it inhibits ribosomal biogenesis. The first is that PRMT5, as I mentioned, is important for methylation of SM proteins, which do splicing. And if you look at the effect on splicing with these inhibitors, you see a very strong increase in intron retention. And intron retention essentially leads to nonsense-mediated decay and loss of these transcripts. And these transcripts aren't just global, all transcripts, they are specific transcripts, which are very important in the growth signal. So in the mTOR pathway, many of the genes which are important for that seem to be inhibited. The second is specific methylation of proteins which do mediate protein translation. So here's a really nice example of PABP1, which is the protein which binds to the poly-A tail of RNA. And we can show that this PABP the methylation is prevented by PRMT5, and it actually inhibits the ability of it to bind to RNA. Again, another mechanism of how it inhibits RNA processing and protein translation. And then finally, PRMT5 has two very important ribosomal proteins which are targets. There's RPS10 and RPS2, and we believe that the lack of methylation or demethylation of these proteins actually prevents the mRNA getting into the ribosome and being translated. So hopefully I have shown you some data to convince you that PRMT5 inhibitors are going to be helpful or useful cancer therapeutic. Like much of biology, it's not simple. There's not some simple single target. It's multiple targets. And I think that our data would suggest that its predominant action is through targeting multiple sites within the RNA processing and protein translation. And as I mentioned, 120 years ago, people recognized that ribosomal biogenesis, or the nucleolus, is a very prominent and important part of cancer. And so I think that therapies like this are likely to be ones which will have this therapeutic window and be able to target fundamental processes that cancer cells depend upon. And at that, I will leave to answer any questions. Thank you very much.